Vanakkam. Welcome to this video on biomechanics. We have been looking at skeletal muscles. Specifically, we are interested in understanding what is a sarcomere and how a sarcomere produces force. So, in this video, we will discuss sarcomeres and how sarcomeres convert excitation to contraction, or how a command coming from the central nervous system is converted into force. So, we continue our discussion on muscles and the hierarchical arrangement of muscles. So, if you take a muscle fiber and if you subject it to light microscope and you view, you will find there are these thick bands. I am discussing the bands that you see for a striated muscle. Skeletal muscle is a striated muscle, just clarification. You will see these bands. So, you will have alternating thick and thin bands like stripes in a shirt, like you have a striped shirt. So, alternating stripes that you will see, these stripes that are seen, since they are present in the muscle, that muscle itself is called as striated muscle. Striated muscles means those muscles that have these stripes. But what is the functional significance of these stripes, of these lines, of these striations, right? So, you will have these thick lines that are there here, here, and then on top in this figure, this is seen in green here, that there are some other relatively thinner filaments that are seen, and it appears as though the thick threads or filaments are hanging loose in air, but this is not the case. You need to just zoom in a little bit to see more details. So, if you take a myofibril, a muscle fiber and a myofibril, so and you zoom in and you see these lines, okay. And what do these lines represent? Remember that at the highest level you have the muscle itself, and this muscle is composed of bundles of fascicles which are further having smaller bundles, and then you have one fascicle which is composed of several myofibrils, and then you have within this myofibril you have this striations, these lines, these zones. So, you are having this unique arrangement in which there are these thick lines followed by thin lines. And the point from which these thin lines originate, from which the thick lines are suspended using a thin, very thin, not visible in this picture, is a protein called titin. Through that protein, from these two ends, the thick filament or the striated filament, or the one that appears as striations, that filament, and the thinner filament are suspended. The point from which this suspending of these filaments happen is called as a Z disc, and the zone between two Z discs, or all the components between two consecutive Z discs, is called as a sarcomere. Sarcomere is this smallest functional unit of a muscle. So, functionally this is how small it can get. So, if you would like to explain the function of a muscle, you start with the function of a sarcomere. Okay. So, the zone or that region or that set of components between two Z discs constitute a sarcomere. This is the smallest functional unit. in a muscle. Let us zoom in a little bit. I am zooming in further. So, you are having these thick filaments and you are having these thin filaments, and I am zooming in. If I zoom in, what I find is that there are these thin filaments shown here as blue and the thicker filaments. These are the thicker filaments. Thick filaments are here, shown in light orange. Yes. These are the thin filaments, shown in blue. Okay. Now, you can see that there is a spring-like material, the protein called titin, right, which is holding the thick filament in space between two Z discs. Okay. In the middle of this, you have an M band, and the region where the thick filament starts and ends is called as a H band. Okay. And the set of all components between two Z discs is called a sarcomere, which is the smallest functional unit in a muscle. Now, what do these thick and thin filaments signify? How do these produce force? This is of interest for us. It turns out that the thick filament has these heads, like small buds in a stem. Okay. These heads are capable of attaching to specific points on the thin filament. Specific points on the thin filament are capable of attaching to these heads, and under specific conditions, this attachment between the thick filament and thin filament will be so strong, and the thick filament will undergo a conformational change, pulling the thin filament, thus making a really minuscule, a very small amount of contraction. We will discuss the physiological details, or we will discuss the functional details of how this happens in a future video, but here we just introduce these two notions. This thick filament is called, technical name, myosin. The thin filament is called actin. Frequently, students will find it difficult to remember this. In the class, in future classes, I will frequently switch between thick filament, thin filament, and myosin and actin. I will keep on interchanging this. You need to remember which is myosin and which is actin. Simple way to remember, actin is thin. Actin rhymes with thin. The other one is myosin, the other one is thick. Okay. Actin is like thin. Okay. The other one is thick. This is how you need to remember which is which. Now, what are the conditions that lead to the situation in which this force development happens? First of all, before this, we need to understand how and where the command comes from and how this command is processed in the muscle cell. Somewhere in the central nervous system, a decision is made, likely the frontal lobe of the brain, not necessarily always the frontal lobe of the brain. So, there is this question, not all movements are voluntary, maybe some of the movements are being generated only at the spinal cord, maybe it is a reflexive movement. Without going into the details of what this is, somewhere in the central nervous system, a decision to make a movement has been made. We will just say this for the purpose of this discussion. And this is communicated to a muscle through a neuromuscular junction. For the purpose of this course, we will not get into the details of how the neuron and neuromuscular junction functions, because we restrict our attention to the biomechanics part. So, if you are interested in understanding the neuroscience of how this happens, I request you to check my other courses or the other videos in which I have discussed neuromuscular junction, how the decision is converted into a command and how that is being communicated. That I request you to please check in YouTube or just Google, you will be able to find that. So, we restrict our attention to how the command that is received from the neuron is processed in the muscle cell. We are interested in how the muscle cell is converting this electrical signal that is received into a mechanical output, which is this phenomenon in which an electrical signal is converted into a mechanical output is called excitation contraction coupling. Excitation means something that is electrical in origin. Contraction means is the result, right? This is the cause, this is the result, right? So, this excitation is in electrical form and the output is contraction. We are interested in understanding contraction because contraction is a mechanical aspect. We are interested in biomechanics of how this happens. But first, there must still be some processing of this electrical signal. How is this happening? That is the question. We focus our attention to that topic in this slide. So, what happens briefly is that a neuron receives and sends a command to the muscle to start contracting. This is received through what are called as neurotransmitter. In this case, this neurotransmitter is called acetylcholine. This is a bit of a detail, but better to start here. This neurotransmitter is received by the receptors or a special kind of channel that is present in the muscle cell plasma membrane. This channel is sensitive to acetylcholine. So, whenever acetylcholine is present in its vicinity or when it is present or attached to a specific region in that channel will open and let in a lot of sodium. This sodium that is coming in will be so high that it will change the membrane potential in that region and cause a chain of events or the so called domino effect. It will cause a chain of events that will lead to an electrical pulse that is getting generated here. This pulse is called an action potential. So, this domino effect, a lot of sodium enters inside because the command has been received through chemical means. Remember, acetylcholine is a neurotransmitter which is a chemical. And this leads to a chain of events or a domino effect whereby electrical signal or a particular pulse with a specific characteristic is generated, that is called action potential. This action potential has a tendency to travel along the plasma membrane of the muscle cell, because neurons and muscle cells are excitable cells, and because muscle is also an excitable cell, it has this potential and the tendency to conduct electricity, conduct this action potential. So, if a pulse is generated at one part of the muscle cell, it gets transmitted throughout the rest of the plasma membrane. So, this pulse will go in this direction. So, it will be felt here and then it will be felt here. Actually, it will be regenerated at each point, but that is a matter of detail. I request you to check in other videos. So, this pulse is coming through this, and it turns out that the nature of the muscle cell is such that every now and then there are these transverse tubules that you see in the muscle cell. These transverse tubules are such that it resembles the English capital alphabet T, right. It goes like this and then takes a dip and then goes back like this. That is the nature of this T tubule or the transverse tubule. Going forward, I will only use this, call this as T tubule. So, these T tubules are present, which also conducts this pulse, and the nature of this T tubule is such that very close to it there are these so called sarcoplasmic reticulum. This is the equivalent of the endoplasmic reticulum, but found in the muscle cell. Endoplasmic reticulum in the general animal cell, when specialized to muscle cell, is called as the sarcoplasmic reticulum. It turns out that this sarcoplasmic reticulum has some channels that are attached on two sides, one on the sarcoplasmic reticulum itself and the other, because the T tubule is very close, the other end of this the other end of this channel is attached to the T tubule. Okay. And the nature of this channel is such that whenever it detects an action potential, whenever it detects a pulse, an electrical pulse traveling at the T tubule, it will open. And when it opens, it also opens its partner on the sarcoplasmic reticulum side. So, it is like two doors. When one door opens, the other door also opens. The first door here is on the T tubule. That first door is called as the dihydropyridine receptors, or in short DHP receptors. These DHP receptors are such that whenever it detects the presence of an pulse or an electrical activation or an action potential, it will open. And when it opens, it also opens the other door that it is connected to. That other door is present not on the T tubule, but on the sarcoplasmic reticulum. That other door or that other channel is called ryanodine receptors, or RYR. So, let us review this one more time. So, whenever the command is received from the neuron, an action potential is generated in the muscle cell, and this action potential has the tendency to travel along the length of the plasma membrane of the muscle cell, and as it is traveling, it will also go through the transverse tubule or the T tubule, and in the T tubule you have two receptors. Actually, in the T tubule you have one receptor that is sensitive to this action potential. Whenever there is this action potential, it will open, but when it opens, it also opens a door or a channel that is attached to the sarcoplasmic reticulum. Sarcoplasmic reticulum is not on the plasma membrane. Sarcoplasmic reticulum is in the cytoplasm of the muscle cell, or rather the sarcoplasm of the muscle cell. What is the difference between cytoplasm and sarcoplasm? Cytoplasm in the general animal cell, when we apply to the muscle cell, is called sarcoplasm. So, the sarcoplasmic reticulum is in the sarcoplasm of the muscle cell. It is not on the T tubule. So, it is like this. There are two consecutive doors. The first door opens whenever there is an electrical activity, but this door has the tendency to open another door on the other side of the corridor. There is a corridor, there is a small gap in between. So, you see this is the corridor that I am speaking about. This is the corridor that I am speaking about. So, there is one door which will open whenever there is an action potential, but this door has this tendency to open the other door or door on the other side of the corridor, which is on the sarcoplasmic reticulum. What is so special about this sarcoplasmic reticulum? It turns out that the sarcoplasmic reticulum is capable of storing a large amount of calcium. And it turns out that calcium is very crucial for conversion of force, for production of force. Why is this? We will see in a future video. But for now, let us just assume that we need a lot of calcium to produce force in a muscle. And all this calcium is collected and stored inside sarcoplasmic reticulum to be released only when the command is received from the neuron. Command is received from the neuron, action potential is generated in the muscle fiber, and it is traveling through the T tubule, and it is detected by the DHP receptors, and when the DHP receptor opens, it also opens the ryanodine receptor on the sarcoplasmic reticulum. This is where we are. And when this receptor opens, when the door on the other side of the corridor, when that opens, so this is the corridor that I am speaking about. There is a door here, there is a door here, and there is one more door here. This is extracellular fluid. This is sarcoplasm. This is the sarcoplasmic reticulum. Whenever the action potential reaches along the plasma membrane, and this is the plasma membrane or the sarcolemma. Okay. Plasma membrane of the cell. So, whenever the action potential reaches, it opens this DHP receptor. This door is called as DHPR. This door is called as the ryanodine receptor. Whenever the DHPR is opened, it has the tendency to also open, because it is not like this. There is a connection between the two through the sarcoplasm, such that whenever DHP receptor opens, it also opens a ryanodine receptor that opens the gate through which calcium can leave. Because of the nature of the fluids within the cytoplasm, it turns out that, or in any fluid, if there is a semi-permeable membrane, there will always be exodus of particles from a region of high concentration to a region of low concentration, something that we know. And because a lot of calcium is stored inside the sarcoplasmic reticulum, and the rest of the cytoplasm is maintained practically free of any calcium, there will be a huge concentration gradient between the sarcoplasm and the sarcoplasmic reticulum. Remember, both of these are within the muscle cell under discussion. Both the sarcoplasm and the sarcoplasmic reticulum are within the muscle cell, the same cell. But when the sarcoplasmic reticulum opens, because the concentration gradient is so huge, when it opens, a lot of calcium leaves from the sarcoplasmic reticulum into the sarcoplasm. This situation, in which there is a sudden buildup of a large amount of calcium within the rest of the sarcoplasm, not counting the sarcoplasmic reticulum, is called calcium spark. Okay. This situation is called calcium spark. The buildup of, suppose I were to plot the amount of calcium as a function of time, let us say this is time in milliseconds or microseconds as the case may be, and this is the, let us say, concentration. In the y axis I have concentration of Ca2 plus. You will have a baseline concentration which is very close to 0 actually. This will vary, let us say that is 0, slightly above 0, or maybe you have this, and then there is a sudden increase of calcium that happens. There is a sudden spike that happens. This spike is called calcium spark. its role is so functionally crucial for the production of muscle force. We will see why in a future video. For now, it is sufficient for us to know that this build-up of calcium or this maintaining of calcium concentration gradient is crucial for the production of this calcium spark and other after effects of this calcium spark. But why do this? How do you do this? First of all, how do you do this? Because now that I have said that there is this calcium spark, that means that calcium is leaving and spreading in the rest of the cytoplasm. Once that is spread, how will it come back to the sarcoplasmic reticulum? Because once it is not like one muscle is a use and throw muscle, single use muscle. It is not like that. Once it contracts, again it will have to contract within a few milliseconds again. So that means that there is a need to maintain the calcium within sarcoplasmic reticulum, bring the calcium back from the rest of the cytoplasm or from the sarcoplasm back into the sarcoplasmic reticulum. How is this achieved? Through a mechanism called calcium pump. This is a matter of detail. Without going into much detail, I will try to very briefly explain what this is. So whenever you need water that you need to pump from the sump or from an underground well, for example, maybe a bore well or a real well or whatever, or from a sump, you want to pump it to a overhead tank. Why do you need that? So that you will have what is called as running water, right? You open the tap, water will come in the tap. How does it come? Because there is water in the overhead tank, and because we live in planet Earth and there is gravity, when you open, water rushes in from the overhead tank to your tap. Water is always running. One of the reasons why we waste water. Suppose you had to carry this water from ground floor to the place where you live, we would not waste water. Anyhow, that is a distraction. Coming back, so I open the tap, water comes. For me to do this, first of all, I will have to pump the water from the sump to the overhead tank. How do I do that? I use a pump, right? Water pump. I spend electricity. It's an electric motor that pumps water from the sump to the overhead tank. Because this pumping has to happen against the gradient, against gravity, I will have to expend energy. I pay for this. Right? Electricity, electrical energy is expended to pump this water from a region of high concentration, from a region of essentially from a low height to a higher height. Let us just say that from a lower level to a higher level against the gradient. So whenever you want to push something against the gradient, you have to spend money or you have to spend energy. Right? That is what we do. So we are expending electrical energy, we pay for this in rupees, dollars, whatever, we pay for this, we expend energy, we send the water from the sump to the overhead tank. Similarly, suppose I would like to add calcium to the sarcoplasmic reticulum, which is already having a large amount of calcium. This is against the gradient. Suppose there was an opening, suppose there was a channel that allows passage of calcium, naturally calcium will go from the region of higher concentration, which is the sarcoplasmic reticulum, to the region of lower concentration, which is the rest of the cytoplasm or the sarcoplasm. So if there is this channel that generally allows calcium to leave, calcium will be leaving from the sarcoplasmic reticulum to the sarcoplasm, because in general sarcoplasmic reticulum has a lot of calcium, right? This is the same situation that happens when you open the tap. Why does water come down? Because there is a gradient difference, right? From a higher potential energy to a region of lower potential energy. I am just opening the tap, water comes. When the water does not come, that means that there is no more water, or you are at the same potential energy, perhaps that, right? So for you to push the water up, you need to expend energy. Suppose I want to push calcium from the sarcoplasm to the sarcoplasmic reticulum, that is from a region of low concentration to a region of higher concentration, that is against the gradient, like you are pushing against gravity, against the gradient, you expend energy through in the form of ATP, you spend energy and pump this calcium, find wherever the calcium ions are and then push them inside the sarcoplasmic reticulum. There must be some very good evolutionary reason for this to happen. There must be some very good evolutionary reason. There better be a good reason for us to do this, right? Why would you do this? Because you have to expend energy. Normally, this is not needed. Why do you even need this? No, such is the importance of calcium in this function. So, you need to spend money, or in this case energy, to push this calcium inside the sarcoplasmic reticulum. Maintenance of this huge concentration gradient between the sarcoplasmic reticulum and sarcoplasm is extraordinarily crucial for the function of the muscle. Because of this reason, the system goes to extreme lengths in terms of spending the energy to maintain this concentration gradient between the sarcoplasmic reticulum and the sarcoplasm, such that any small opening in the channels in the sarcoplasmic reticulum will lead to a huge buildup of calcium because of the great difference in the concentration gradient, leading to this calcium spark situation. You want this calcium spark situation for muscle force production. But how is the sarcoplasmic reticulum able to store so much calcium? Because this is a bit of deep physiology, so we will spend some time on this. Because the sarcoplasmic reticulum has some special proteins such as calsequestrin. Calsequestrin is this special protein that has the ability to hold anywhere from 18 to 50 calcium ions. It can hold and absorb up to 50 calcium ions. A single calsequestrin molecule has the ability to hold on to 50 calcium ions. With the use of such special enzymes, this sarcoplasmic reticulum is able to hold on to a huge amount of calcium. Right. So, through this pump and through the functions of these proteins like calsequestrin, it is holding on to a large amount of calcium. So, this is how calcium is maintained within the sarcoplasmic reticulum, and whenever the command comes from the neuron, the action potential is generated in the muscle cell plasma membrane, and this travels along the plasma membrane and it reaches the T tubules and it opens the DHP receptors, which further leads to the opening of the ryanodine receptors on the other side through what are called as foot processes. Foot processes of the DHPR. Through those, the ryanodine receptors open. That is the opening that calcium was waiting for. A lot of calcium is present inside, and all that calcium, or a large amount of that calcium, will suddenly rush out. A huge outflux of calcium, not outside the cell, within the cell. All this happens within the muscle cell. From within the sarcoplasmic reticulum to the sarcoplasm, there is a huge gush of calcium. Right. This leads to a situation in which calcium is suddenly becoming abundant in the sarcoplasm, and this calcium and detection of this calcium happens in the muscle fiber at the thick thin filament interaction level, leading to a situation in which contraction happens. Right. So, this is how the excitation that happens in the muscle cell, or rather in the neuron, there is this excitation which leads to this chemical being released, which then regenerates an excitation in the muscle cell, is converted into contraction. Exactly how this contraction is developed is something that we will see in the future video. Okay. So, a large amount of calcium is stored within the sarcoplasmic reticulum, and this is open through the action of this action potential through the interaction of the DHP receptor and the ryanodine receptor, leading to a situation in which a huge amount of calcium leaves. That is called calcium spark. As an aside, something that we also want to look at is the special case wherein sometimes what happens is there are some channels in some muscles, there are some channels that are sensitive to action potential, okay, that are present in the T tubule, that has a tendency to let in calcium from the extracellular fluids. These are the special L-type calcium channels that open whenever the action potential reaches. These are depolarization sensitive, or the action potential sensitive channels that open and let in some calcium inside. And there are some more channels on the sarcoplasmic reticulum that are sensitive to the presence of calcium, which will open. So, there are two channels, one here and one on the sarcoplasmic reticulum, through which calcium enters, and calcium sensitive channels are also present on the sarcoplasmic reticulum that detects the presence of calcium outside the sarcoplasmic reticulum or in the sarcoplasm. So, whenever calcium comes from outside the cell to inside the cell through this L-type calcium channels, it detects and the sarcoplasmic reticulum opens, leading to a situation called, and when that sarcoplasmic reticulum channel opens, it releases a lot of calcium. The reason why it releases a lot of calcium is because it detected the presence of calcium just outside the sarcoplasmic reticulum. So, it releases calcium from the inside. So, this situation is called calcium induced calcium release, or rather CICR. In the good old days, it was thought this is how calcium is released from the sarcoplasmic reticulum. From the outside of the cell, calcium is entering, and that opens the calcium channels from the sarcoplasmic reticulum. But now it is understood that this happens only in a specific kind of muscle cells, but not in all the muscle cells. In particular, the first method that I described, which is the DHP receptor ryanodine receptor interaction, is how the skeletal muscles lead to calcium spark. But then where does this happen? The calcium induced calcium release. The calcium induced calcium release is known to happen predominantly as a predominant factor in the cardiac muscle. Slightly different from the focus of this course. We are interested in the skeletal muscles. CICR is something that is more pronounced in the cardiac muscle. That does not mean that it does not happen in the skeletal muscle. In the skeletal muscle, the most important mechanism by which this calcium is released is through the interaction of the DHP receptors and the ryanodine receptors. Okay. So, with this we come to the end of this video. In this video, we discussed what is a sarcomere and how excitation coming in from a neuron is converted into the buildup of calcium, which will then further lead to a contraction in the muscle. Thank you very much for your attention.